A single GPCR can turn an outside message into very different instructions inside a cell. The outcome often depends on which heterotrimeric G protein the receptor activates. Three major pathways repeatedly tested in cell biology and pharmacology are GS, GI, and GQ. GS usually raises cyclic AMP. GI usually lowers cyclic AMP. GQ usually raises cytosolic calcium and activates protein kinase C. Let us build all three pathways from the same starting point and then compare them side by side. A GPCR is a membrane receptor that crosses the plasma membrane 7 times. Its ligand-binding region faces the extracellular space while its intracellular loops contact a G protein. The G protein contains one alpha, one beta, and one gamma subunit. Beta and gamma remain tightly associated as a beta-gamma complex. The alpha subunit belongs to a family such as G-alpha-As, G-alpha-I, or G-alpha-Q. In the resting state, GDP is bound to the alpha subunit. The resting heterotrimer sits near the inner membrane surface and commonly held there by lipid anchors. When an agonist binds, the receptor changes conformation. The activated receptor then acts as a guanine nucleotide exchange factor or GEF. It promotes the release of GDP from G-alpha. Cellular GTP enters the empty nucleotide binding pocket. GTP binding changes the shape of the alpha subunit. G-alpha GTP and beta-gamma can then separate or rearrange, allowing both components to contact effectors. The effector that is regulated depends mainly on the alpha subunit family and on the cell type. The signal continues only while the alpha subunit carries GTP. G-alpha hydrolyzes GTP to GDP using its intrinsic GTPase activity. Regulator of G protein signaling or RGS proteins can greatly accelerate this hydrolysis. G-alpha GDP then reassociates with the beta-gamma and restores the resting heterotrimer. Begin with GS, in which the letter S means stimulatory. A GS-coupled receptor activates G-alpha-S through the same GDP-GTP exchange cycle. G-alpha-S GTP binds Adenylyl Cyclase on the cytosolic face of the plasma membrane. The Adenylyl Cyclase used by this canonical pathway is a membrane enzyme, not a free protein floating from the cytoplasm. Activated adenylyl cyclase converts ATP into cyclic AMP or CAMP. Because one active enzyme can produce many CAMP molecules, this step amplifies the signal. CAMP is a soluble second messenger that moves through local regions of the cytosol. One major CAMP target is protein kinase A or PKA. In inactive PKA, regulatory subunits hold the catalytic subunits in an inhibited complex. CAMP binds to the regulatory subunits. These release active catalytic subunits. PKA phosphorylates specific serine or threonine residues on the target proteins. Rapid targets can alter metabolic enzymes, ion channels, or contractile machinery. PKA catalytic subunits can also act in the nucleus and promote phosphorylation of CREB. Phosphorylated CREB works with co-activators to regulate this transcription of selected genes. GS can therefore produce both rapid cellular responses and slower changes in gene expression. Beta-1 adrenergic receptors in the heart commonly couple to GS. Their CAMP-PKA signaling increases pacemaker activity and strengthens contraction through downstream targets. Glucagon receptors in the liver also couple primarily to GS. They promote pathways that help release glucose during fasting. Beta-2 adrenergic receptors can activate GS in airway smooth muscles. Their CAMP-dependent effects favor relaxation, showing that the same messenger can produce different responses in different cells. Now, turn on GI, in which the letter I means inhibitory. A GI-coupled receptor activates G-alpha-I by replacing GDP with GTP. G-alpha-I GTP binds adenylyl cyclase. Unlike G-alpha-S, it inhibits the enzyme. Less ATP is converted into CAMP. The cytosolic CAMP concentration falls relative to the stimulated level. PKA activity consequently decreases. Proteins that would have been phosphorylated by PKA remain less phosphorylated. GI therefore often opposes a GS signal that converges on the same adenylyl cyclase CAMP system. However, GI signaling is not simply an off switch. The released G-beta-gamma complex is itself an important signal transmitter. In cardiac pacemaker cells, G-beta-gamma from M2 muscarinic receptors opens GIRK potassium channels. Potassium conductance increases, making the membrane potential more negative and slowing pacemaker firing. G-beta-gamma can also inhibit certain voltage-gated calcium channels in neurons. This can reduce calcium entry and decrease neurotransmitter release. M2 muscarinic receptors use GI signaling to slow the heart. Some GI family proteins also regulate additional enzymes through G-alpha or beta-gamma signaling. The third pathway is GQ, which primarily signals through phospholipase C-beta. A ligand-bound receptor activates G-alpha-Q by GDP-GTP exchange. G-alpha-Q-GTP binds and activates PLCβ at the inner membrane surface. PLCβ acts on phosphatidyl inositol 4,5-bisphosphate or PIP2. Inositol 1,4,5-triphosphate or IP₃ is soluble and moves through the cytosol. Diacylglycerol or DAG remains embedded in the membrane because its fatty acyl tails are hydrophobic. IP₃ binds IP₃-gated calcium channels in the endoplasmic reticulum membrane. These channels open and calcium stored in the ear lumen flows into the cytosol. Cytosolic calcium can bind proteins such as calmodulin and trigger many responses. At the plasma membrane, DAG cooperates with calcium to recruit and activate conventional protein kinase C isoforms. Activated PKC phosphorylates target proteins on the serine and threonine residues. GQ signaling can therefore change secretion, contraction, metabolism, and gene expression. Alpha-1 adrenergic receptors commonly used GQ in vascular smooth muscle, where increased calcium supports contraction. M3 muscarinic receptors use GQ in glands and smooth muscle to produce cell-specific secretory or contractile responses. Now compare the three pathways on one screen. GS stimulates adenylyl cyclase, raises CAMP, and usually increases PKA activity. GI inhibits adenylyl cyclase, lowers CAMP, and usually decreases PKA activity. GI-derived beta-gamma can independently regulate potassium and calcium channels. GQ activates PLCβ, splits PIP2, and generates IP3 plus DAG. These are signaling tendencies rather than universal outcomes, because some receptors couple to more than one G protein family and each cell expresses a different set of downstream targets. Each pathway also contains mechanisms that end the signal. G-alpha hydrolyzes GTP, stops regulating its effector, and rejoins beta-gamma. Phosphodiesterase convert CAMP into 5' AMP, terminating signals that raise CAMP. When GI switches off, Adenylyl Cyclase is released from inhibition, allowing cellular CAMP control to recover. After GQ activation, ACRCA and other pumps remove calcium from the cytosol while IP₃ and DAG are metabolized. GPCR kinases and arrestins can also reduce further coupling of an active receptor to G proteins. Two bacterial toxins reveal the opposing logic of GS and GI. Cholera toxin ADP ribosylates G-alpha-ase and prevents efficient GTP hydrolysis, leaving GS signaling persistently active. Adenylyl cyclase then remains stimulated, causing abnormally high CAMP in intestinal epithelial cells. Pertussis toxin ADP ribosylates G-alpha-I and prevents productive activation by receptors. GI can no longer inhibit adenylyl cyclase normally, so CAMP may rise in affected cells. When solving a pathway question, first identify the alpha family, then the effector, the second messenger, the kinase, and the final response. Remember the core comparison. GS raises CAMP, GI lowers CAMP, GQ generates IP₃, DAG, calcium, and PKA signaling. One receptor begins the message, but the G protein helps decide which intracellular route opens.